Thank you, John. And let me say it's wonderful to be here and to see so many participants in UK Biobank and so many researchers. I just wanted to start this session with a personal thanks to all of those who've taken part in Biobank. It's an incredible enterprise and an amazing data set, as I hope you'll walk away today thinking. I also want to, again, personally acknowledge how much effort has gone into it by the many, many people who are working on the Biobank team and who do such a terrific job. We're going to talk about imaging now, which has been one of the most recent enhancements. And let me just kick off with imaging of the brain, which I'm going to highlight how important, even though fortunately most of the people who are participating in Biobank today are healthy, how important this investment is for helping us all to understand diseases of the brain. Biobank is very special because it is not just about the brain, it is about the brain in the context of the body and exposures, lifestyle, a great deal about each of the participants. This is critical data because what we are beginning as neurologists and neuroscientists to struggle with is understanding how environment and lifestyle, the exposures, what we might call the episome, is influencing the brain through life. We may be born with a susceptibility to a late life disease like Alzheimer's through our genetics, but not everyone who has the genes for Alzheimer's succumbs. There is something about the exposures and the way our lives are lived that makes the disease start to express itself. Moreover, the special feature of Biobank is that we are studying people through their lives, well before they ever develop disease. That's critically important. Because what we as neurologists are beginning to learn is that disease starts very, very long before it's ever clinically manifest, sometimes decades before. This is an example for Alzheimer's disease. This is a crude measure of time. Just think of time increasing over here. These are measures, so-called biomarkers, that are telling us something about the underlying pathology in the brain, what's happening behind the scenes, as it were. Now, what I want you just to focus on is at this time, whatever it might be, that's when someone actually starts to develop Alzheimer's disease. This is when they start to complain of memory problems. But what's really important is, although this is a somewhat arbitrary axis, we now know that the time at which the first changes can be detected in people, in this case measuring special proteins in the fluid that surrounds the brain, literally decades before the disease ever comes. There's only one way to observe these disease processes before they become expressed, and that is to do population-level studies of the comprehensiveness of Biobank. Now, moreover, we all are becoming very, very aware that it's a funny thing, but for a very long time we thought that the brain was sitting up here in a cavity, somehow protected from the rest of the body and disconnected. Now, of course, that's an impossible thought. What we now appreciate is that the problems of the brain are the problems of the body influence the brain, and vice versa. So the diseases that we succumb to systemically, in this case like diabetes, have huge influences on the brain and on the risk of these late-life brain diseases. For example, here's an illustration of the way people are thinking about metabolic syndrome and the development of type 2 diabetes, its relationship to cerebral vascular disease, and the way in which early vascular disease may trigger the early processes of Alzheimer's disease to lead to cognitive decline. This all becomes a vicious circle. So we need to understand what is happening to the brain and what is happening to the brain before the expression of disease in the context of everything that we're seeing in the body. Again, this is what a study like Biobank is needed for. And finally, I've talked a lot about disease, I've talked about biomarkers, which are proteins and things that you can measure. But of course, disease doesn't happen to tissue, it happens to people, and particularly for the brain. The way in which disease is manifest, the time at which it is first noticed, its impact, all of this depends on not just on the biology, the cellular mechanisms and neural networks, but also on the way an individual is relating to other people, relating to their society, whether they're employed or not employed, what they're doing, what activities they engage in. These are things that have been out of bounds for many disease-specific studies because doctors and scientists have traditionally focused on the biology. But resources like UK Biobank become a treasure trove because those of you who have participated have been so generous in sharing a rich amount of data about what your lives are like. Now, the imaging enhancement, which will involve about 100,000 of the people in UK Biobank by its end in 2023 or so, is an extraordinary enterprise. I will confess that when Rory came to me 7-8 years ago and suggested it, I was very skeptical. I thought it would be I thought imaging was something that you could only really do in a specialized place in the hospital. What Biobank has shown is that you can take imaging out to the field, you can make it into a process like any other test, and you can do it more efficiently and at higher quality than we would have been able to do otherwise, even if we had been able to do it within a hospital. What is being done in Biobank now is imaging of the head with state-of-the-art techniques, imaging of the heart and the body, which you'll hear about after me, DEXA imaging of the bone and ultrasound. And all of these are being done on the same people at the same time, allowing these data to be interrelated. Let me just give you a flavor for what you can begin to see with this. For the brain, basically this is a bit simplifying it, but 3 types of imaging are being done. Imaging of brain structures, seeing what is where and how big things are in the brain. Imaging of the wiring diagram of the brain, the way in which these areas of the brain all are connected to each other by the nerves that run in the deep white matter of the brain. And then imaging of the functions of the brain, as measured by functional magnetic resonance imaging, to look at the way in which these areas of brain are talking to each other. Now, the imaging of the structure, as illustrated here, is for the hippocampus. If you kind of blow this up and are somewhat imaginative, you can see that what we're really looking at, and a great deal of correlative work over the years between histopathology and imaging has emphasized this, is when we look at changes in brain structure, we're really looking at changes in brain cells and their density, the nerve cells in the brain. So it's giving us a really deep window into what's happening deep inside. When we look at the functional images of the brain and the ways in which different brain regions are connected to each other, we're actually getting a surrogate, an alternative marker for a great deal about the underlying molecular structure of the brain. This is a functional image from UK Biobank showing how this area of brain tends to talk to this area and this area. This is from something called the Allen Brain Atlas, in which each of the bright areas tells us about a cluster of genes that is expressed in the same way in a particular part of the brain. So it's telling us about something that's happening inside cells between genes and proteins. And what this is telling us, I'm sorry, it's a bit faded here, there's a brain on the outside, is these are areas of connection between the brain determined from this sort of map and this sort of map linked together. It's telling us that this kind of map is actually giving us evidence about the underlying gene changes within individual cells in the brain. So we can see structure and function. They give us a huge window on the brain. What can we do with this? Let me just give you a few illustrations. The exciting thing is it's happening now. The data is already being released. This was not done with UK Biobank, but it kind of gives you, I think it's one of the most dramatic examples. This was a tiny study. This was only 5,000 people, aggregated together from many studies. Biobank's already 3 times that. And here, this is a map showing us the size of brains in healthy people over these age ranges. And this shows us a group of people who have a range of diseases. And what is important is right here. Here is age, proportions survived. And what you can see is that if your brain is more robust than the average, the age at death is late. If the brain appears less robust by these measures, conducted from the brain scans, earlier death is predicted. So mortality seems to be tightly related to these metrics from structural brain changes. And UK Biobank also is incredibly rich in brain. It's beginning to show us that changes in the brain really are related to lifestyle and the environment. I'm just going to give you a flavor of this very quickly. This is data from Biobank that Carla Miller and her colleagues, along with many of us, put together last year. This is representing the 11,000 or so variables that all of you contributed to UK Biobank about your thinking, your general physical health, your lifestyle. What this is showing, the higher along this axis a point is, the more significant the association between a change in this brain structure and these factors is. What it's telling us that there are lots of highly significant associations, telling us that there may be some relationship between measures of, in this case, physical activity and brain structure, and similarly for other things. This is telling us the same sort of thing. These are measures of functional connectivity, the sort of functional imaging I showed you before. These are showing us that functions of the brain also are closely linked. There's too much data there to begin to decipher for you, but I'll just leave you with a final note as we move into the next lectures, is that it's, of course, not just about looking at the brain in isolation. The whole point of doing all of this imaging is to be able to relate the many, many things we can learn about the body. These are images of fat, which Jimmy Bell will tell you about more. This is where fat is in the body, and we can begin now to probe the relationship between fat in the body and fat in the brain, which is carried in the insulation of the wires around the brain. And finally, look at these in the context of disease risk factors. Finally, I want to emphasize that the pioneering efforts of UK Biobank to make all of this data available to researchers worldwide is catalyzing the integration of other data sets with it to further increase the value. And this is, for example, from the Dementias Platforms UK, where the UK Biobank has been joined together with other large cohort studies across the UK, many of which are disease-focused, so there becomes a link between what's happening to everyone in UK Biobank and to the disease, in order to really drive future dementia research. And finally, I just want to close by saying how many people have been involved worldwide in setting this up. This is a landmark project. It's doing extraordinary things, and I just want to emphasize 125 or so other people really contributed selflessly to set this up as a research resource for the world overall. Thank you very much.